Hello everyone, welcome to our second in our autumn webinar series of ease. I'm just going to allow a few minutes for everyone to join the room. I can see the room is filling up already, so thank you for being patient in the waiting room. Can I just ask you, make a note in the chat just to say who you are and where you're calling us from, where you're joining from today, what time of day it is, because I'm conscious that for poor Elisabeth, who has joined us today, it's very early in her morning. So we thank her for getting up early and propping herself up with a cup of coffee to make sure that she can be on top form for our webinar. So for me in the UK, it's lunchtime, but I know we've got people joining from right across the globe. So please just pop in the chat where you're calling from and say a quick hello. Just give a few minutes for everything to catch up. Greetings to everybody. So there's people coming in from Poland and Amsterdam and the UK, Croatia, more people from the Netherlands. So lots of people coming in. So good welcome to all of you. So I'm just going to share my screen for a moment. So we're delighted to have with us today Dr. Elisabeth Bik. And as I say, she's in California, so she's very early in her morning, but we're pleased to have her with us today. And it's been a great honour to have Elisabeth, because obviously she's a very busy lady, and I know lots of you are really looking forward to seeing what she has to say and hearing from her. So that should be a really good session today. So we're going to start off with just a quick introduction from me. My name is Mary Hodgson. I'm the Secretary of EASE. And for many of you I know aren't EASE members, it's really great to have you along to one of our events. So we run these webinars. We run 8 of them a year, a session in the spring and then another session in the autumn. And so we've got 8 of these events going on throughout the year with guest speakers, and it's a really nice way to keep everybody up to date with the latest thinking in particular topics. So that's our speaker for today, Elisabeth. So as I say, introduce yourself in the chat once we get going. As you go through the presentation, if you could type any questions that you have into the Q&A, and then just use the chat for maybe some comments or some useful links if you've got things that that you'd like to tell other people about. And then just to let you know that the session will be recorded and it will be publicly available on our EASE YouTube channel as soon as I can sort of render the video after the event. And then people who are in different time zones or people who are just busy at this moment will be able to catch up with the recording. And please, I'm going to send you the recording, all of those of you that have registered. So please feel free to share that with your, your colleagues and spread the word as far as you can. That would be great. Okay, so let me just tell you a little bit about Elisabeth. Elisabeth is in Holland. She's a biologist living in California, and she's worked for 15 years at Stanford University and 2 years in the industry. So since 2019, just before the pandemic, she has been working primarily as a sort of volunteer science integrity person and an occasional consultant. And doing that, she scans the biomedical literature for images or other data of concern and has reported over 4,000 scientific papers. So she clearly has huge experience. So Elisabeth is the founder of the Microbiome Digest and the Science Integrity Digest blog, and I'm sure she'll refer to those as she goes through her presentation. She was awarded the 2021 John Maddox Prize for outstanding work exposing widespread threats to research integrity in scientific papers. So lots of other accolades to her name as well, but that's just a quick introduction from me. So without further ado, Elisabeth, I'm going to stop sharing my screen and allow you to say hello and to share yours. I'll see you straight after your presentation.
Great. Well, thank you, Mary, for the introduction. And people were asking, or somebody was asking in the chat, which if they can tweet about it and which hashtag to use. Would just hashtag ease or ease event or something like that, would that be appropriate?
I will pop all that in the chat right now.
Okay, great. Thank you. And then with that, I will share my screen. And so you should see my screen. So yeah, I'm sort of a microbiologist turned science integrity warrior, or whatever label you want to put on me. I've had many. I'm on Twitter, so my microbiome digest without the E here is my handle, sort of reflecting my older microbiome days. as I was entering social media and starting Microbiome Digest. But currently I'm most active on Science Integrity Digest, which is the blog where I occasionally will talk a little bit about certain cases I found or certain concerns I share. And I also have tips there how, for example, to report plagiarism, or how to which software to use for image detection, software that is freely available, because most of the readers of my blogs are interested in that. They cannot afford big, expensive packages. So my disclosures first, I occasionally work as a consultant, not very often. And that would be for universities or publishers, editors, to investigate particular cases of allegations of misconduct. Obviously, I cannot work on the cases I've discovered myself, but I serve sometimes as a second opinion, and I get paid as a consultant for that. I also get speakers fees to give talks. And I'm also on the editorial board of eLife, which is also, I receive an honorarium for that. Most of my income, however, is through patreon.com, where people can donate small amounts of money, like €1 or $5 or things like that, to support my work. That's an account where a lot of makers, people, for example, who write books or who draw cartoons, get funding like through online supporters. So it's a sort of a donation site, and I get a lot of income, and most of my income currently from that. I also have worked at a company called Biomedical. That company started to do some shady things, and I had left the company by then, but they were raided by the FBI, and the founders, not the employees, but the founders have been charged with insurance fraud. And so I wasn't involved in that. I was the scientific editor, but yeah, this will be, of course, on my resume, and a lot of my the people who do not appreciate my job will continue to bring up that I worked at a fraudulent company. But again, the founders, but not the employees, have been charged with the fraud. They're also fugitives, so it's going to be hopefully a movie one day, because I think it's a great story to share. There's a lot of people who do what I do, which is post-publication peer review. We look at papers and we might find problems in them that have not been caught during peer review for whatever reasons. And most of us specialize in something. So I specialize in image duplications. And the people whose name here are in boldface are also doing that same work. Some of them operate under pseudonyms, as you can see like Smut Clyde or Cheshire or Morty are not their real names, but they're, yeah, they operate more in the anonymous. And Claire Francis, I think, is the most famous one of all. That's a person who is using a pseudonym and also has found a lot of image problems. But others are focusing more on things like duplication, plagiarism, citation problems, things like statistical errors, ethical concerns, all kinds of things like that. Or they provide the platforms to write about these cases or post them on papier. So of course we all share the same ideology. Like for me, at least, science is about finding the truth, and publications are the way we communicate with each other. So we care about publications because that is what other scientists will base their work on. We all build our work on the work of others. We're sort of layering new layers of bricks in the wall of science, and every brick might be a publication. But if some of those bricks contain errors or fraudulent data, that means that other scientists might spend a lot of time trying to replicate those results, but cannot do so because of these errors or potential fraud. And so we care about publications because we feel they need to be flagged if there's a problem with some of these publications. Now, most scientists are honest, and we all have maybe somewhat naively assumed that everybody is honest, but there is fraud in science. And in the US, at least, and I believe also in many other countries, science misconduct is defined as one of three things: plagiarism, well, I think we all know what that is, copying somebody else's text or ideas without giving credit; falsification, where a person does an experiment, but changes some value. So it becomes from a negative a positive, or maybe leaves out an outlier, sort of making the results look a little bit better. And fabrication is where a person completely makes up results. And that is, of course, the worst kind. And fabricated results, if done smartly, can look very realistic. So why would people cheat in science? Of course, the pressure to publish, the publish or perish atmosphere in science has only increased. As scientists, we all feel that pressure to publish, and it's much easier to publish positive results than negative results. And this, I think, is a thing that should maybe be improving in scientific publishing, because in many cases, negative results are as valuable as positive results, but they're much harder to publish. And so it might be tempting to make the results look a little bit better and get them published. And of course, there's different scenarios. So maybe a person has been very successful early in their career and now moved on and is not very successful. He or she might be tempted to cheat. And also there's a lot of hierarchy in academia where a person who is a professor or a group leader has a big position of power. And if that person is not a good mentor, but a bully and very demanding of their staff and their students, they might actually threaten to fire a junior person if they don't give the results that he or she had been hoping for. And these threats can be so big that a person feels the need to cheat, otherwise they cannot get a paper. And so I usually talk about this because I feel there's behind every case of misconduct, there's a sad story. And it's very easy to point fingers and yell fraud. And I try not to do that. I try to avoid that because there's always a drama or a sad story behind the scenes. So I usually make it about the papers, but not about the people, because it can be very time consuming to figure out who the guilty person is. And I sort of rolled into this work because I found by accident image duplication in 2 chapters of a PhD thesis that had plagiarism. And by accident, I saw that these figures contained images that had been reused to represent different experiments. And in one case, this image with a very distinct little spot here was used twice, and one time in mirror image to represent different experiments. Excuse me. And so I realized, I guess I have some talent to find these duplications, because these PhD chapters had also been published went through peer review and had been published. And I wrote to the editors, and these got fairly quickly retracted. And so I thought maybe I should look if I can find more of these examples in scientific papers. Now, scientific papers, at least in the biomedical field that I'm familiar with, might look like these. There's lots of types of images, and roughly you can divide them in line graphs on the left and photos on the right. And the one, of course, the duplication I detected was a photo. And we humans are very good in spotting that, for example, all these bands in a Western blot look different, and we can tell them apart. And same for bands or lanes in a gel, or photos of tissues or mice or cells or whatever. We might be able to spot all these images are fine, but we might be able to spot overlaps or duplications. Well, in line graphs, it's actually very hard to detect that. And any person who wants to fabricate, it's much easier, obviously, to fabricate a line graph. Like, who knows if this graph was really a measurement? Now, I have no reason to assume it's not. Like, I do think these are fine experiments, and actually 2 of these images are my own. But it would be very easy to fake something like that, to make a fabricated graph. While with photos, at least you maybe, with the current state of digital photography, we can still distinguish real from fake. But at the end of the talk, I will talk about if that's really still the case. But let's assume for now we can detect duplications in photos better than in line graphs. And so there's several types of duplications that I found along the way. I started screening images from scientific papers that had photos, and I would find roughly 3 types of duplications. So here on the left, we have a simple duplication where there's lots of panels. Each of these panels is a different experiment, but 2 panels marked here by me with red boxes, and here 2 panels marked by me with blue boxes, look identical. So the exact same photo has been used twice to represent two different experiments. So it's inappropriate. Of course, sometimes images can be duplicated appropriately, but this is inappropriate. These should not have been the same photos. And it's also very likely that such simple duplications are honest errors. People just make tons of photos and maybe they didn't label very well, so it's sloppy. But it's not necessarily misconduct. And I want to stress that some of the duplications I'm finding are, maybe even half of them, are not misconduct, are errors. Here in the middle, we have an example of a repositioned duplication. Here we see 4 photos representing, they have different labels, 4 different experiments, but these 2 overlap here at the top. And these 2 here at the right also overlap, also with some change in the aspect ratio. And so this, is this an honest error? If you do 4 experiments, you would expect 4 different non-overlapping photos. And at best, we're looking at 2 samples. And yeah, so, this is a little bit more likely to have been done intentionally than the simple duplication. And these repositions could also involve mirroring, like the one I showed you in the previous slide, or flipping, rotating, shifting, changing aspect ratios, etc. Here on the right, we have a duplication with an alteration, sort of a maybe convoluted term where we try to refer to photoshopping. Basically, in this photo in panel A, 2 lanes, lane 1 and lane 3, marked here with blue boxes, look the same to me. They look identical. And in panel D, 3 lanes look identical. So here we are looking at duplicated elements within the same photo. And that is most likely to have been done with the intention to mislead, because these things do usually not happen by accident, although in certain cases there might be technical explanations for it. But basically these division of these 3 different types of duplication helps a person in determining whether or not it was likely that it was done by error or by misconduct, with the caveat that the middle position can be both. And of course, there might be technical explanations, and sometimes you can have a paper with lots of duplications in the simple category, so many that the paper, in my opinion, should be retracted because it just goes beyond, an oops, I made a mistake. It's just like so many that that paper could be retracted. But I feel it's a little bit helpful to make a judgment on the severity of the allegations of misconduct. And of course, these duplications can sometimes also be visible in line graphs. There can be simple duplications, 2 times the same image. There can be stretched. In this case, the same plot was stretched. And there can be duplicated parts. In this case, it's not duplications within the same graph, but across graphs, there have been duplications. There's many other examples. Here is a particular example where this is a type 1 duplication. There's different experiments, different treatments, but these 2 photos are identical and these 2 are too. So this is a simple duplication, and I feel this should be addressed with a correction. I don't think this was done intentionally. I would have done it better, but if I really wanted to cheat. But in this case, there's a I assume they made the figure layout and they just forgot to add these 5 micromolar photos, for example. So I wrote to the editor in October 2015, but unfortunately, this journal never took any action. And this paper is still out there with its duplication. And of course, it's not the end of the world. I assume if people try to replicate these papers, it would still work. But I don't know, maybe it's my sense of perfection, but I feel this should be addressed with a duplication. But the journal did not take any action. Here's another type 2 duplication with a repositioning. So we're looking here at 4 photos of kidneys of, I forgot if it was rats or mice, being treated with different compounds or different concentrations. And these 2 photos show a small but significant overlap. So instead of 2 different treatments, these photos were taken from the same specimen, just moved under a microscope a little bit. And again, I reported this in October 2015 when I started to do this work, not full time, but like really only in the weekends. But I started to report a lot of these cases to the editors. And this particular duplication has still not been addressed, and it's been a couple of years. This is another duplication, a type 2 duplication, and this is an example where there's so many type 2 duplications, so many overlapping images that it goes beyond a little, a little error that could be addressed with a correction. And here the journal retracted the paper, and I think that's a good outcome. There's so many overlaps that you cannot really trust these results anymore. And but it still took a lot of years. Like I would look at this image and think this is an easy decision to retract this paper, or at least do something about it. But I'm personally a little bit frustrated how long this took. I had to send several emails to PLOS ONE to remind them that I had reported several papers to them and that they should look at it. Here's another example of an extreme case of type 2 duplications, lots of them. And this paper also got retracted. And this one was fairly quick. I reported in February 2015 and I got retracted in October 2015. And I think it's a good outcome because I would not want to base my experiments on this particular paper or this particular figure. So I think that's a good outcome, obviously not for the authors, but for the readers in general. Here's a case of duplications with repositioning that actually got retracted yesterday. So this is a case I reported in July 2014, so 8 years ago. And finally it got retracted. And I'm not quite sure why that took so long, because in some of these cases, 4 images are all from the same sample. These 4 are all from the same sample, these 4, and even though they are representing different experiments. And so I don't think this should have taken 8 years. I mean, this is like, for me, a 5-second decision, but I'm not quite sure why this took so long. And maybe we can discuss that in the discussion later on. Another case of a repositioning. Here, there's lots of duplications here, where the same sample, for example, I mean, all these experiments are different mutants, I think, being tested. These are photos of plants and plant leaves. And for example, there's an overlap between this one and this one with some rotation. And there's so many overlaps that I feel that is beyond what could be addressed with a correction. And again, opinions might differ on that, but I don't know, that's a lot of errors to make. And this one got corrected. At least there's some action taken, but I'm not quite sure if that's the right decision to address this problem. And then moving on to type 3 duplications. Here are photos of fungal spores. And in this particular paper, there are several duplicated elements in panel B and in panel D. There's several duplications here marked with rounded boxes of different colors. And this paper got corrected. Here, I mean, this for me is a clear case of photoshopping, and this should not be addressed with a correction according to COPE guidelines, but it was, and the authors actually were allowed to send in a new version of panel B and D, and it got corrected. Here's a case of severe image duplication type 3, where it looks like parts of these bands have been duplicated across, in different parts of the figure. There's lots of areas that look duplicated to me. I actually reported this first only online, but I also reported it to the journal, but no action yet. So this one is still pending, but it's relatively not that long ago since I reported it. Here's a case where these things also happen in big impact factor journals. So this is a paper in Science from a pretty famous person. Has been a lot of very active in politics and was on TV a lot, but there's a duplication between these 2 panels. And this paper, first Science didn't really respond, but finally they retracted it a couple of years after I reported it. Well, there's some intense cases of type 3 duplications. These are nanoparticles, but there's so much duplication going on here that I almost get dizzy looking at it once you see it. And this took several years to get retracted. Excuse me. Again, this is a I don't know, a 5-second retraction for me, but it took many years and a lot of frustration to get it retracted. These are flow cytometry images where parts of these panels appear to have been duplicated. And again, this paper got retracted. So applause one, yeah, still took, 7 years almost to retract this paper. And then this one of my favorites, if you're familiar with the Surgisphere retractions, that was a set of papers in the New England Journal of Medicine and the Lancet, I believe, that claimed that hydroxychloroquine had lots of side effects and was not helping with the COVID-19 treatment. That paper used a large database collected by the Surgisphere company. And it turns out that lots of these parts of these patients were or appear to have been made up, and the papers got very quickly retracted. But the founder of Surgisphere, so presumably the person who fabricated this data, he generated this figure in as part of his PhD project, so many years before the COVID-19 epidemic, and it looked like, so these look like Melba toast to me, but they're actually parts of inner ears of rodents, and parts of these images look duplicated. Here's some close-ups of the chinchilla and the tree squirrel here. And there's just many duplicated parts, at least it looks to me. And this, in my opinion, cannot be explained by a technical error. But the journal allowed here for a correction. The last author, the PhD mentor, wrote to the editor having a very vague excuse about layers and duplications. And to me, it just sounded like, I don't know, like made up. But yeah, the journal allowed for a correction. I feel this is the complete wrong decision. It sends a very wrong signal to the reader. And these corrections for image manipulation make me very angry. And yeah, I feel that needs to be said. So I'm going to fight this with COPE. I have very low expectations, but I hope that to get this retracted, because I feel if we allow people to cheat, like this person has cheated multiple times, papers got retracted, and he caused in the end for a lot of confusion. I feel this is done at the early stages of his career. And yeah, it sort of shows that in some cases, people keep on cheating throughout their career. So I did a more scientific approach of trying to determine how often these cases of image duplication would occur. So what I did is I did a systematic search of 40 different journals from 14 different publishers to see if I could find multiple cases of image duplication. And as I showed you previously, I did find many of these duplications. I scanned all of these papers by eye. And so I probably have missed a lot, but I also wanted to make sure that I didn't falsely accuse anybody of an image duplication that was inappropriate. So my two co-authors, Arturo Casadevall and Ferric Feng, had to both agree with my findings. And if one of them didn't agree, we took out that paper. So they were very experienced. They were editor-in-chief of journals, and so they had seen a lot of cases of images and editorial decisions. They were very experienced in that. So we collaborated amongst the three of us in what to call inappropriate image duplication. And in that set of 20,000 papers, and again, I think a lot of people do not believe that I can scan that many, and I was still employed at Stanford, so it was sort of a hobby. But if you only focus on the images, you can go through most of these papers within a couple of seconds. And sometimes it takes more time to download all these papers than to actually assess them and analyze them. So we found 800 papers, 4% of these papers to contain duplicated images within the paper. And again, if the paper did not contain a photo, we didn't include it. And if one of us didn't agree with it, then we also didn't include it. But 800 papers passed that filter, and that's 4% of papers with photos. These were mostly molecular biology photos or papers. So mostly Western blots, images of tissues, of cells, flow cytometry images, which are technically not photos, but we also included them. And of course, not all of these are misconduct. Some of these might be simple duplications or just one set of two images that overlap, which is probably not intentionally done. But we made an educated guess that about half of these papers, so 2% of all the papers I'd screened, contained intentionally misleading duplications, as we usually word it. Now, of course, that doesn't necessarily mean that all papers, so not just looking at molecular biology papers, but all kinds of papers, is that 2% true for all papers? Now, we realized that if a person is a very good Photoshopper, or if you move your sample a little bit farther under the microscope, it wouldn't overlap anymore. But also you could very easily fake data by typing in numbers and making a beautiful graph. That's fabrication, and it's actually surprisingly easy to do. So we do think that the real percentage of misconduct is much higher than 2%. It might be, in the 5% range or 10%. And of course, it's hard to ever get a good estimate of how many papers contain misconduct, but that we feel that it should be higher than 2%. That's our best guess. Because we looked at 40 different journals, we could also look at the impact or the correlation between impact factor and the percentage of problems in these journals. So each dot on this graph is a journal from a different publisher. They're colored according to publisher. And you can see here the impact factor on the x-axis, and that's a log scale. So here we have Science and Nature and PNAS. And there was one journal, the Journal of Cell Biology, that has very strict image rules and has been screening their journal for a long time for images upon exception of manuscripts. So they had already a very good screening factor in place, but I did find one paper in a set of over 100. So they ended up the best. They have the lowest percentage of problems. And some journals with low impact factor did really poorly. So there seems to be sort of an inverse correlation, but then some journals with low impact factors did really well. And these are society journals. And yeah, it looked like some journals outperformed other publishers. But it seems to be not really publisher related. It's more maybe on the individual level of editors, and perhaps also correlated to the audience of these journals, like a society journal or a very specific molecular biology journal type of journal would have maybe a different audience and maybe more knowledge of the authors who send in papers and maybe more scrutiny. I'm not quite sure. But yeah, overall, there seems to be a negative correlation between impact factor and problems. Now, of course, because I reported most of these in 2014 and 15, we had a good data set to see what happens to these papers after reporting them to the editor. And I wrote to the editors under my full name. Excuse me. And after 5 years, after reporting these papers, unfortunately, 2/3 of these papers had not been taken action upon. 27% had been corrected, tiny sliver of expression of concern, and then 7% were retracted. But yeah, it's sort of sad to see that in 2/3 of these cases, there was no action taken by the journal. And I waited 5 years. It wasn't, and I sent several reminders. It wasn't that I, demanded quick results. I feel 5 years is a very generous time to expect an outcome, but yeah, no action. And yeah, I feel that's a very sad state, and we can discuss this. I can think of several reasons. Maybe editors have no idea what to do with these things, or they don't feel supported by their publishers. Maybe authors have moved on and are hard to find. Maybe the corresponding email address doesn't work. Maybe the author threatens to sue the editors. I've heard of cases of that. Maybe the editors are waiting for institutional investigations. But as Holden Thorpe, editor-in-chief of Science, recently wrote in a column, like maybe we should not wait for institutional investigations. These take forever. And it's part of that is trying to figure out who was responsible for making a figure. And usually data is lost, especially if a paper is a little bit older. people point fingers, people are threatening with lawsuits, and these cases can take forever. But maybe a journal should take action before these institutional investigations have panned out, because you can see these problems separately. The problems in a paper can be viewed at separately from the question of who did this. You have a problem, and you could retract the paper without waiting for an institution to finish up their investigations, and that might speed things up. I'm not quite sure if there are objections to that, but I feel that might be a good solution, because quite frankly, this is a sad state of affair, in my opinion. So because I tried the official way, and also sometimes contacted research integrity officers of universities and did not receive a lot of response there, I have posted most of the problems. By now I have posted over, I think this is getting closer to 6,000 of the papers that I found on pubpeer.com, which is a website where you can talk and discuss papers with potential problems. And so there's a PubPeer allows for they have a plugin that will work with your browser. And if you have installed that, then you can see if a paper has received a PubPeer comment. This is, for example, a PubMed search. And so PubMed is a database that a lot of biomedical people use to do literature searches. And if a paper has a PubPeer comment, you will see these green banners if you have installed the PubPeer plugin. And I feel this is currently the best way to warn readers, because if we have to wait for corrections or retractions, as we have seen, these can take many years. And I feel it's in the interest of the reader to know that there's a potential problem. Now, PubPeer is not just, as somebody called it recently, pub smear. It's moderated. You cannot just say this person is a fraud or this paper is all made up. You have to show why you think that's the case without actually saying and using accusatory words. So you can say figure 2b looks remarkably similar to figure 3c with an illustration, or you can say this paper appears to have no ethical permits or so, or maybe it looks like this data was collected before the ethical approval was obtained, or this paper appears to contain text that is the same as text previously published and with some illustration of things. So as long as it's objective and verifiable, you can post it, but you cannot just yell whatever you want. So it is moderated, and I feel that keeps things polite and objective and scientific. Of course, I searched for all of these images until 2 years ago by eye, and but of course you might argue, well, computers can do it better than a human, and I agree with that to some extent. And I'm starting to use software, and people have developed software to find these duplications, but it's much harder than you might think. I've participated a couple of years ago in a DARPA challenge, and there was a media forensics challenge. And so I had my data set with duplications and non-duplications. And it was very hard for the software developers to develop tools to find these duplications. And there's many reasons for that. But now, a couple of years later, of course, AI, artificial intelligence, is going to solve the world, right? And so AI can be used to develop software to spot these duplications. And so I feel the current software tool that is used or starting to be used by several publishers is Proofig, developed in Israel. I unfortunately have not been given access to the tool, so I have no idea if it works or not. And I'm a bit mad and sad about that. I have seen cases where I see a duplication and Proofig used by an editor is not seeing it. Sometimes these overlaps are very small in the corner, or with a little bit of rotation, they might look slightly different and not be identical on the pixel level. So I have dealt with editors who say, well, we used Proofig and it didn't find the duplication, so you are wrong. And that's incorrect, because we cannot rely on the software yet. It's not there yet to be better than a human. Like, I do feel we're still at the stage where humans are better, but there are also occasions where the software, I'm using Image Twin, which is made available for me, but it is a commercial product, where Image Twin has found many more duplications than I could find, but it also flags a lot of false positives. So you still need a human to assess the results. And there are many cases where it just flags too many things, but occasionally there is a duplication also between papers, because it also has a database of figures from other papers. So it occasionally will find duplications across papers that I would have never been able to find, just using my eyes and brain. And I've been quite impressed with Image Twin, but the false positives are causing a lot of extra work, because you have to check all of them to see if they're real or not. But of course, artificial intelligence can also create a lot of problems. So it can create fake images. And these are images created by the random face generator. So these people do not exist. So you have to go to thispersondoesn'texist.com. And every time you hit refresh, it will make a new photo. And these photos are artificially generated. These are not real people. They're created out of databases of photos, but these are completely fake and pretty much indistinguishable from real. And of course, there's DALL-E, which is also AI, but it's a slightly different technology, where images are created also on databases of libraries of images, but these are text descriptions. So it will just, you just type in cells under a microscope and it will come up with images that look surprisingly real, but are completely fake, but they're unique. So they would never be detected with software that would focus on duplications across or between papers or within papers. And so these images look pretty realistic. And if you think about, how realistic dinosaurs now look in movies, I'm a bit pessimistic about the future, because AI can just generate fake images that look very believable. And it can also create text that looks very believable. And so how can we know for sure that future submissions are written and generated by humans as opposed to technology? And of course, there's the big problem of paper mills. I don't have time to go very deeply into this, but these are commercial companies that generate completely fake papers, or that generate authorship positions and sell authorship positions on papers that have already been accepted. And in some cases, these have been found to work together with editors on special issues who get some kickback of the profits of these companies. But these companies, so there's different models, but they're usually active in countries where there are very strict requirements about publishing. For example, China has a regulation that if you are about to finish medical school, you have to have 1 or 2 papers published in scientific journals, even though these are clinicians who are not interested in research and who are not given time to do research. So they're faced with an impossible requirement, and they will buy an authorship on these papers. And apparently these companies, paper mills, advertise freely on social media in these countries to have to buy a paper. And so they offer these papers. And there are some of the ones that we looked at in China, for example, the several Chinese paper mills, they have usually authors from China, they have shared authorships. Usually all the authors have no publishing history. And the data can look surprisingly real, but the photos are sometimes real, but they're often reused across papers. So you might find 2 papers from a completely different set of authors with overlapping images, suggesting that they have a common source. And the papers are usually written on some template, but the type of cancer is changed to something else, or the type of RNA molecule is changed to something else. And I have to give credit to all these people here, because I didn't work on this by myself, but this was a group effort, and several people have found hundreds and probably thousands of these completely fake papers. And then, there's also legal threats that I'm dealing with. Of course, people are not happy when I talk about, when I criticize their papers, and I can see that. But there have been 2 cases, and this case in particular, I want to point out, where the leader of an institution in France has threatened to sue me because I criticized a lot of his papers on Papier. There were papers, this is a person who has published a lot, and several of his papers appear to miss ethical permit statements, so doing human research without a proper ethical approval, without IRB approval or consent. There were cases where the same approval number was used for dozens of papers. And initially that might have been a correct approval number, but you cannot reuse an approval number for different types of research. And that appears to have been the case. There have been papers I found by this lab with duplicated images, and I showed one of them in the previous slide. And yes, there's lots of problems that appear to have been associated with this lab. And all of these concerns could easily be taken away if he had shown ethical permits, or if he had shown the original images, for example. But instead, he threatened to sue me. He made several videos about me, which have thousands or millions of views on YouTube. Published several statements on the website of the institution, and luckily there was an outpour of support for me. People started petitions to sign to support my work, because we all feel that these cases of scientific criticism should not be held in the courtroom. And I hope this will never lead to a real legal case. It's just a threat at the moment. But in the end, this work could financially ruin me. I'm not really, I'm supported by Patreon, but I don't have large amounts of money to defend myself if I'm being sued. But I'm willing to take that risk. In most of these cases, these are just threats, but of course they can go to something bigger. But I'm willing to go that route, and I feel very supported by a lot of the scientific community. So this is my last slide. How can we prevent misconduct? And of course, we're never going to prevent it. People are going to fraud, but we can make it a little bit harder. I feel that the recent open science or good research practice movements are very good developments. Basically, how a journal could do it is having strict requirements for example, showing original photos of Western blots and original data for original measurement for values that are reported as mean with standard deviation. You could ask for a little bit more data, and a scientist should have all of that. It's still, fresh and warm by the time they submit the manuscript. We should somehow focus less on publications and productivity as a measure of productivity, and maybe have more openness and willingness to accept negative results, and not only focus on novelty, but also focus on reproducibility. Because in the end, if we can reproduce a paper, maybe that's the only way we know it is real and not fake. There's lots of cases where I feel there's conflict of interest. I've seen cases where the senior author on a paper was also the editor-in-chief of the journal. And, that can happen, but it makes it very hard if a paper needs to be corrected or retracted. There's a clear conflict of interest there. And these papers usually do not get retracted. And of course, institutions have similar conflicts of interest, because they might not want to admit that there was misconduct within their walls, or they might not want to fire the person who brings in a lot of grant money. I do hope we can move towards much faster investigations. I feel 5, 6, 7, 8 years is way too long to address these concerns. In the meantime, these papers can be cited, and people might try to reproduce them and waste money because of that. Maybe there should be, I don't know, like a 6-month or a 12-month limitation to these when an outcome should come. And we should also have more practical guidelines for when to retract or correct a paper. I feel the COPE guidelines are not really good enough, and maybe we can work on that. People like me, we can be called troublemakers or whistleblowers or fact checkers, depending on how you feel about this work. But I do feel there should be some pay involved and protection, legal protection. And I hope that publishers will move towards hiring people who are specialists, who might look at images the way I look, but also screen accepted manuscripts for statistical problems, for example, or ethical concerns. And because peer review is not really designed to detect these problems, and this is work that is specialistic, and it should be paid, I hope. We should also, to punish the fraudsters, just at least retract the papers, or maybe have them lose their job, because I feel if you cheat in science, in my opinion, you're not worth to be a scientist. I've touched upon artificial intelligence. It can solve some of the problems, but it can also definitely create more of them. So I'm not sure how to handle that. And in the end, there's a tremendous cost of misconduct, not just for the reader, for other scientists, but for science as a whole, because you could argue that any retraction will decrease the faith that the general audience have in science. But on the other hand, it could also increase the faith, because you can show that science is being retracted if it's bad. And but of course, in the end, it should not have been published in the first place. You could argue that. But yeah, we need science to be, solve all the problems in the world. And I firmly believe in science. And I do not want anybody to walk away from this talk thinking that all science is flawed, because that's not the case. There is, most science is good. And of course, there's errors, but we need science to be also a little bit more self-correcting, because only then we can use science to deal with the problems that we face on this planet. And with that, I play the game Image Forensics on Twitter, so you can spot the duplications in these panels. And I do this to make people more aware of the problem, but you can also earn an emoji award if you're the first person to solve the puzzle. So with that, I will stop sharing and hope that there are many questions.
Well, many thanks, Elisabeth. That was an amazing talk. Thank you so much. And yes, there are many questions. So let's kick straight off. We have lots of questions here. So here's one from Kathy Sigel. She says, some authors take offense to your comments on PubPeer, and I hope that this is not the norm, but what percentage of authors are eager to offer an explanation when you comment on their paper?
That's a good question. I would not actually know. I have no good way of measuring that, because I cannot mine the database of PubPeer. Excuse me. But very, very few authors will comment. And if they comment, the comments can be ranging from a good explanation. There have been a couple of cases where I'm right in seeing a duplication, but it was actually the same experiment. It was just labeled very differently. And so I just didn't understand that. And I love to be proven wrong, but that's very few cases where that happens. Yeah, some authors are immediately just yelling back at me that, accusing me of all kinds of things. And that, taking away from the problem itself. And I feel those people might actually be very guilty, because they don't have any way to prove that I was wrong. They immediately attack me. Yeah, so very few authors actually come back, and I try to tag all of their email addresses if I can find it. And I hope that we can have a dialogue, but many choose to not respond. And I would say like 80% or so, if I had to make a wild guess, is not responding.
Okay, thank you. So here's another question from Emily Arturo. She asked, how should a journal use apparent intent of the authors in deciding whether to correct or retract? And she then follows up with, how does one deal with COPE directly, as you say you have done, to address an issue that the journal is not addressing?
Well, yeah, it is hard to know if there was intent and to mislead. So I think you could also just look at, even if there was an intention to mislead, but you see 10 overlapping panels, it would, in my opinion, deserve retraction, because the data is just not reliable. And a lab that produces so many errors and so many overlapping images, you could also question the remainder of the data in those papers. So whether or not there's an intent to mislead, in some cases that is obvious, but in some cases it's just too many errors to save the journal, to save the paper, and it should be retracted. So it is a hard question, but sometimes I've seen retraction where, yeah, it's not based on an intention to mislead, but based on just too many errors in one paper. And the second, I was trying to find where is she?
It was about COPE. It was about your COPE.
Right. Okay. Yeah. Yeah. To be honest, my experience with COPE is very frustrating, and of course, COPE is run by editors, and so it's not going to be in my favor. In most cases, COPE will decide that the journal has handled cases appropriately, as in they did contact the authors, the author offered an explanation, and so they thought that was satisfactory and did not address the problem. But I'm looking at these images that might look very Photoshopped to me, and I don't find the explanation of the author very satisfying. And I often don't even get to see what the author wrote me or which original photos they send me. And I've seen cases where the authors will send in so-called original that are also clearly Photoshopped, but I'm not offered to look at these. Obviously, I'm a party, but like I feel in many cases, COPE will decide that the case was handled appropriately and no action was needed. But I'm not convinced of that. So I have not had a good outcome, but I'm still protesting the cases where I feel it was mishandled, because I feel, yeah, I mean, somebody should object to that. But yeah, it's sometimes hard to fight these decisions.
Sure. Okay. So Anne Cambon-Thomsen is asking, do you contact only editors or authors or both when you find an error or duplication? Like, do you start with one and then the other, or is it a mix?
Well, a couple of years ago, I would just write to the editors, and that's, I thought, the most appropriate way, like a dialogue with the authors, usually, even especially if held in private, is not leading to anything constructive, because the author is going to defend themselves, attacking me. So I just wrote to the editors and let them handle it. But yeah, I was frustrated with the lack of response in most of these cases. So I'm just posting on Papier, and Papier will grab the corresponding email address from the PDF and will send an email to the author. And I try usually to add more email addresses if I can find them. I mean, in some cases it's hard to find the email address of a person. So currently the authors are notified and not the journals. And of course there's advantages and disadvantages in that. So the authors do not see my email address. They can only reply through the Papier system in public. And so it is contacting the authors, but it keeps things a little bit more polite. But then the problem is that the journal might not know. So Papier does offer, I think they call it a dashboard, where if you are an editor of a journal, you can have like a subscription to Papier and get notification if there's papers flagged from that journal on Papier. And but I'm not part of that. If a journal decides to not screen Papier, then they might never know that there's a comment. And so occasionally I will send to the editors as well.
Sure. Okay, so there's a bit of a follow-up on that. So Leonard Schalkwyk, I can't pronounce that very well, so excuse my pronunciation. He said, Papier has had a big and positive role, but is there a concern that we are dependent on this single organization? It does place some limits on how the database is used.
Right. Yeah, Papier is run by volunteers. So I feel currently the best system we have, with some continuity over the past, I think it's like, I don't know, 8 or 10 years old. There have been other platforms trying to log comments, but I feel this is currently the one that is the most central, the most wide, ranged. But yeah, I mean, it's limited because of course the Papier volunteers are hoping to make some money out of it, but it is run by volunteers. So it's not, it has flaws, but I feel it's the best system we currently have. I would not know of any other system that is as widely used as, for example, comments on a particular paper. I've commented on several papers on PLOS ONE and the comments just disappeared after a couple of months. So I'm frustrated about that because they're gone. And this one at least is a little bit more stable.
Okay. So Ángeles Oviedo-García asks, he has 2 questions to ask. So if the editor are involved in, for example, citation rings or reviewer rings or coercive citation, where could be sent misconduct allocations? COPE does not seem to be very helpful in this.
Right.
And
so
I don't know if this is, he follows up with, how do you report misconduct if the cheaters are colleagues from your own department or faculty? So I suspect he's got something going on there.
You're right. I mean, yeah, I usually get a lot of questions about like very personal situations, and I don't want to go into much detail about that. I mean, it is a problem. If editors are cheating themselves, how do you tackle that? I've dealt with several cases where there were multiple papers by the same author, and that author was an associate editor on several journals and an editor-in-chief of one of them. So those papers are getting away with very mild corrections, and I tried to raise that with Cope, and Cope said it was all handled appropriately, and I think we agreed to disagree there. It is very hard. I mean, sometimes I try to deal with them, with the publishers. I have some good contacts at several publishers, but they also don't seem to have a lot of power. So there are lots of these situations where everyone is sort of stuck. I think a lot of people agree it's not a good situation, but it's very hard to get these papers rolling. So there's still lots of conflicts of interest that are hard to break through. And Cope cannot really deal with that. I don't think it's set up for that. And it's, in the end, I feel always going to choose for the editors, not for the whistleblowers. And in case of personal situations, I would say that is a very tough situation, because you might have inside information that nobody can see from looking at the paper. And so it's very hard to raise concerns if you have inside knowledge, because it's going to out you as the whistleblower, because very few people had that knowledge. And so it had to be an inside person. I mean, the best you can do is remain objective, take your concerns to your either your ombudsperson or your research integrity officer at the institution, and hope they take it seriously. But unfortunately, in many cases, whistleblowers are not believed and are seen as troublemakers, because they use sometimes very emotional language, because they're so involved and so angry about things. So the best you can do to help yourself is to remain very objective and gather as much evidence as you can. But it's very general advice.
So I'm conscious we're going to run over our time a bit, but as we've got so many questions, I'm happy to go on a bit longer.
I'm fine too. I'm fine too.
Sure. Yeah. Okay, so Nicole Beauchamp has asked, with the increased pressure to deposit data into open repositories, does it worry you that it will lead to more fraud, there being a bank of images to manipulate? Oh,
that's a good question. I mean, there's already so many images to take and reuse, and so we need software to crawl these databases and use that as a reference database to screen images from newly submitted manuscripts again. So already we have PubMed Central, which is a huge database that is mined a lot. The images are open access and are mined a lot for software to look at all kinds of things. So these are a rich source of potential reuse. So those are the databases that some of these tools have. And I think we should then extend those databases so that the software tools also include those. But I do feel, I mean, it's not going to prevent fraud, and people are going to find new ways of fraud. We're always a step behind, I feel. But it's sad if you think about it. Yeah, there's so many ways. Okay. So Iva Grabarić Andonovski, she's that EASE Council member, has asked, do you think that the responsibility for detecting image manipulation lies with the reviewers or solely with the authors, solely with the editors? With the reviewers or with the editors? Well, there are cases where I feel that should have been caught by the peer reviewers. I mean, they should spend a couple of extra minutes in a multiple hour peer review to just look at the images. But I don't expect people to pick up on some of the more intricate duplications that I have spotted. I have more experience than I would assume the general peer reviewer. I do hope to raise awareness by posting these examples on Twitter so that people can see them and get a little bit more used to them and be able to spot them as peer reviewers. But I do feel that some of these are too hard to catch by a general person, and they should be caught by the publisher or the editorial office or whatever, but like a paid person who specifically looks at papers only for that reason. And some journals and publishers have have paid staff. Jana Christopher, for example, is a person who's very good in finding duplications, and she has worked for EMBO and currently for FABS. And so there are people hired to screen these papers, but it depends. Not all editors or publishers do that. But I feel it should be a paid position. Yes. Okay, so let's just take a couple more questions.
So we have one here from Seyed who says, do you think code sharing and sample data sharing with licenses helps to stop this kind of fraud, or some expert review the code data image before the final decision? So you need that expert first.
Can you repeat the question? I don't see it. Like I always need
the last comment.
Last question.
In the chat. Yeah.
Oh, in the chat. Okay, sorry.
Code sharing and sample data sharing with licenses help to stop this kind of fraud, or you need some expert review of the code data image before the final decision?
Well, I don't have any experience in codes. I mean, for me, it seems that sharing your original data and codes are a good thing and will prevent fraud, but I'm not quite sure if it could also enhance it. But yeah, I feel sharing is always good because it makes it easier for others to reproduce what you have done. But I have seen cases, this is not for code sharing, but for image sharing. I've seen a case in Nature where the original data was published, so the uncropped gel, and it already had duplicated areas that were like easy to find by software. Like it was very clearly duplicated, and this was the so-called original image. So I feel that even if you require people to submit their raw data, it allows still for people to cheat. It just makes it a little bit harder because they have to cheat a little bit better. But yeah, if people are going to want to cheat, they will do that. And so we can never completely prevent it. I'm not sure if that answers completely the question, but
okay, so let's take this last question then. So this is from someone who doesn't give their name, but says, are journals notified when papers are put out on PubPeer? Do you find that this makes journals more likely to retract the paper if the paper is on PubPeer?
Yes, there have been. Right. Yeah, there have been people, to answer the last question, there have been people looking at whether there wasn't PubPeer, but they posted publicly. So if the concerns were posted publicly on a website, this was sciencefrauds.org, which had to be taken down because of legal implications. That was Paul Brooks who had that, and he had looked at his data set, and he found that papers that had been published online had a higher chance of being either corrected or retracted than papers that he had not posted there yet. I haven't done that myself, but I do feel if a paper is on PubPeer, it will have a higher chance of being addressed for whatever reasons. Maybe, fear of maybe I don't know, the feeling that now it's in the public, we need to address it.
Well, it's about being watched, isn't it?
Yeah. Exactly. Yeah. And so the other question, do journals get a notification? Yeah, some do. So journals or publishers, I'm not quite sure how that works, have to pay PubPeer to get that notification. So that's sort of their current business model. The papers themselves are free, but there's no automatic way for me as a whistleblower or poster there to automatically include the journal. I wish there was a button for that, but I cannot make that happen. But a journal can buy a subscription to PubPeer. Yes.
Yeah, sure. Okay. Brilliant. Well, we don't have time to get to all of the questions, but many thanks everybody for putting your questions to Elisabeth, and thank you for Elisabeth for being brilliant at answering them all. So just before we leave, I'm just conscious of just a couple of minutes just before we leave. I'm just going to share with you a couple of things that are coming up soon with ease. So the rest of the autumn webinar series, we've got 2 more left. Our member webinar on best practice in how to raise your paper's impact, engagement and presence. That's in November. And then the last one of the 2022 series is the implications of open science for editors and authors. So those are 2 similar events to today. For those of you that are in editorial teams or are working in editorial office, we are running our editorial school for journal editors. This is running in November. It's a 4-module course with Pippa Smart, who many of you will know. She's the chief editor of Learning Publishing, ex-president of EASE, so very well known to us all. So she's running that in November. And then the other thing I just wanted to raise your awareness to is our Environment and Sustainability Committee are currently working with the HESI Fellows, looking at the UN SDG Publishers Compact, and we're doing a survey which we'll try and get out to as wide an audience as possible to find out, you know, what steps and what progress that editors and journals and institutions are making towards those commitments that are written down in the Sustainable Development Goals. So just 3 things there that are coming up with ease that you might like to hear about. So with that, very many thanks to all of you for hanging on for an extra 10 minutes. It's really lovely to see you. We'll see you all again at the next event, and please look out for the video and for the recording of this session, which will be with you in the next few days. Many thanks all. Bye-bye.